So hello everyone. I'm excited to give this presentation with Joe Guzman, who I've known for over 20 years. Not only do Joe and I share a background in public health microbiology, but we're both passionate about protecting California beaches. Joe is a surfer, and I'm an island girl, originally from Hawaii, so I feel a special connection with the ocean. Okay, so I'm hoping by the end of this talk that you'll have a better understanding of the significance of fecal indicator bacteria in environmental waters, given their limitations as indicators of fecal pollution, the shortcomings of fecal indicator bacteria test methods, biological variability, and the ecology of FIB. I'll be discussing the use and limitations of fecal indicator bacteria to assess environmental waters, and speaking mostly about their use for monitoring beaches, stormwater, creeks, and briefly discuss the use and limitations of the test methods and proposed regulations. Now, our presentation is not focused on how to perform the test methods. If you're really interested, you can find many references on how to do that. Instead, Joe and I are going to be sharing our knowledge and working experience to help you better understand what the presence of fecal indicator bacteria in environmental waters means. There's a lot of misconception out there that all fecal indicator bacteria can cause disease, they all originate from fecal waste, and that somehow we can eliminate or significantly reduce these bacteria in environmental waters forever. Okay, so I want to start with the background on fecal indicator bacteria, or FIB for short. They include groups of bacteria normally present in the intestinal tracts of humans, animals, and birds. The four main groups are total coliforms, fecal coliforms, E. coli, and Enterococci. FIB are used to assess the sanitary quality of water, to assess potential human health risks, identify fecal sources, and to measure the effectiveness of risk reduction actions. So you can see, understanding what fecal indicator bacteria are, as well as their ecology, could have significant implications in terms of assessing water quality. Now, total coliforms were the first group of indicators used for testing. Years later, the EPA recommended using fecal coliforms. And then after, labs used these two indicators for some years, it was found that they were not so great indicators of pathogens. Now, pathogens are the actual organisms that cause disease. So the EPA maybe it was around 86, recommended that we use Enterococci and E. coli. Okay, so fecal indicator bacteria are commonly occurring in water. So the EPA developed water quality criteria, or limits, based on epidemiology studies showing a correlation between certain concentrations of FIB with swimming-related illness rates. So when FIB concentration exceeds the limits at heavily used beaches, health officials will post signs warning the public about possible fecal contamination. And you can see there's two different types of standards, there's what's known as a single standard, and for total coliforms, it's 10,000 counts per 100 ml of water. Fecal coliforms, 400, Enterococci, 104. And then we can also look at the total coliform to fecal coliform ratio, which should be less than 10. Now, another way of looking at the data is to look at the geomean. And this is based on results from at least 5 weekly samples that are collected during any 30-day sampling period. And you see the numbers there below that correspond to the geomean. All right, so first I want to talk about total coliforms. This is the largest group of fecal indicator bacteria. It includes fecal coliform and E. coli. And that's important to understand because I think people tend to think of these groups as being separate, and actually there's many genera and species of bacteria that overlap those 3 groups. So the rationale for using total coliforms was that they're generally present in higher numbers in the intestinal tracts of humans and other warm-blooded animals compared to fecal coliforms and E. coli. So if water is contaminated by fecal waste, total coliforms can be detected even after dilution, whereas fecal coliforms and E. coli may not be detected. The limitation to using total coliforms as indicators is that they're not specific to fecal waste. There are many total coliforms that exist and proliferate in soil and water as well as in drinking water systems. Okay, fecal coliforms. So the EPA recommended fecal coliforms as being more specific to human fecal waste because they represent a smaller group of bacteria compared to total coliforms, and they can grow at higher temperatures, which is more similar to the human body temperature. But then, guess what? Years after using fecal coliforms, we found out that they're not so specific to fecal waste. There are many fecal coliforms that exist and proliferate in soil, water, and drinking water systems. All right, so what about E. coli? Remember I said E. coli is actually a subset of fecal coliforms. Well, EPA recommended E. coli as being more specific because than fecal coliforms because it represents just one species. But understand there are hundreds, perhaps thousands of different strains within this species group. And, okay, so E. coli is a predominant group within the fecal coliform group. And the limitation is that it's not limited to warm-blooded hosts. It's been observed in the feces of reptiles, and there's been recent studies showing that E. coli can grow in the environment on plant surfaces, beach sand, and beach wrack. So what about Enterococci? Well, Enterococci was suggested as being a better indicator than the other indicators because they have a similar or greater survival rate in water, as do some pathogens like your viruses. So in that way, they are slightly better as indicators. However, they're also not specific to fecal waste. They're ubiquitous in the environment. They can be cultured from soil, sand, plant surfaces, and insects. They're commonly found in food and in other human body sites, such as the oral cavity, vagina, and occasionally the urinary tract. Okay, so the good, and I said I was going to talk about the good, bad, and the ugly. So the good thing about fecal indicator bacteria is that they have been reliable for monitoring drinking water, receiving waters after sewage spills, and they've been useful for comparing FIB levels upstream and downstream in streams receiving sewage effluent. Okay, so what about the not so good, bad, and ugly? Well, the question is, have fecal indicator bacteria been reliable for monitoring environmental waters? And for that, we need to look at Bonde's criteria for an ideal indicator. So, do the indicators meet these standards? And this table shows the standards and then the answers on the right-hand side. So, the criteria are that the indicators should always be present in the feces of humans and warm-blooded animals. And yes, that's true. They must not be able to multiply in aquatic environments. Well, no, unfortunately. The next criteria is that they occur in much greater numbers than the pathogens. That may or may not be true. And then lastly, they must be unambiguously identifiable by simple, characteristic, and reliable tests. That's also a yes and no answer. So you can see that, unfortunately, our indicators don't meet the criteria for ideal indicators. And the reason why I went through the whole history and background on indicators is because, as you can see, over the last few decades, we've had new and quote-unquote better fecal indicator bacteria introduced before researching their ecology. And by that I mean understanding the range of sources where these bacteria are, coming from, their habitats, their ability to survive and grow in the environment. And my concern is that in the last decade or so, molecular markers have been introduced as an alternative to the fecal indicator bacteria. However, few studies are being done to address their ecology. It's very difficult to find funding to support studies on the ecology of indicators and molecular markers, but until rigorous scientific studies address these issues, we'll continue to experience challenges relating to their presence in water with human health risks. All right. So that's about the indicators. Now I want to talk about the test methods and the good, bad, and ugly related to test methods. And I'm sorry to sound so discouraging, and I hope I'm not sounding too critical, but I think it's important to understand the advantages and disadvantages of these indicators, as I mentioned, to really understand the relevance of finding fecal indicator bacteria in water, and what do those counts actually mean. All right, so there's two commonly used approaches for quantifying fecal indicator bacteria levels in water, and that is membrane filtration and IDEXX. And they're actually now considered traditional methods because they're culture-based and they're not molecular. All right, so here are the desirable attributes of indicator methods. We want the methods to be specific to the desired target organism, independent of matrix effects. We want them to be applicable to different water types. So, we don't want to see differences based on salinity or turbidity. We want the methods to be precise and reproducible, to have adequate sensitivity, to be rapid, quantifiable, easy to perform, and low in cost. All right. First, I want to talk about the IDEXX method. Now, IDEXX makes test kits for total coliforms, fecal, and E. coli, and Enterococci. And you see the different names of the methods here: ColAlert, ColAlert 18, EnterAlert. Now, IDEXX is widely used because it's simple and it's less time-consuming to perform compared to membrane filtration. So for high-volume test labs, this is the preferred method. And it detects indicators in 24 hours or less. And below in that photo, you can see a picture of the quantitray. And those little tiny squares are actually, you can consider them little wells containing growth media for bacteria. There's an enzyme in there that is specific for the indicator of choice. And so if the indicator is present in the water sample, it will actually use its enzyme to metabolize the substrate in the media and grow and cause a change in color and fluorescence. Okay, so IDEXX, if you go on the IDEXX website, you can see that it will say it's accurate and there's no confirmation needed. But I put a little asterisk there because in some cases confirmation may be needed, especially at sampling sites that have repeated exceedances in the standards. And I'll talk about how you can do confirmation later. Unfortunately, there's no protocol from IDEXX, but if you search the scientific literature, you can find various protocols out there. So as I mentioned, IDEXX, one really nice advantage to using IDEXX is that if you use the quantitray, you can obtain counts up to around 2,400 MPN, or most probable counts of the indicator without doing dilutions. The cost of equipment, the culture media, and labor are higher for membrane filtration. But then the disadvantage to using IDEXX is in the interpretation of the results. Remember that photo in the previous slide? Fluorescence can sometimes be subjective, leading to false positives. Okay, so now I want to talk about membrane filtration. Here you see a figure that shows a water sample basically being poured onto a membrane, which allows the water to pass through while capturing the bacteria on this filter or membrane, which is then transferred to culture media that's designed to detect the indicator of choice. The media is incubated for 24 hours, and then on the next day, the colonies that grow on the media are counted. Okay, in this slide you can see the different methods, the indicators that you can test for using membrane filtration, as well as the type of media. And in that photo you can see there's some very pretty colored pink, magenta, and sort of yellow media there for the three different types of indicators: the total, fecal, coliforms, and Enterococci. Okay, so the membrane filtration method may not be as widely used because it requires a higher level of expertise. It's more time-consuming than IDEXX. There's a higher cost for equipment and media. Problems with turbid samples, which can clog the filter and actually mask bacteria. But then the advantage to using membrane filtration, and this is one reason why public health labs like this method, is because it allows for further characterization of fecal indicator bacteria testing, which I'll describe later, which could be useful for assessing potential source contributions. So by characterization, I mean species identification and even strain or subspecies typing using DNA typing methods. Okay, so some of you have seen me present this slide before in other talks, and that's because my sort of specialty is in speciating Enterococci. So what this figure shows are the plates of MEI agar on the left, and those little blue dots on the culture media are Enterococci isolates. And that word isolates is important because the way technology is advancing, and you may have heard of whole genome sequencing, the isolates can be used for whole genome sequencing and DNA typing. So that's what we mean by isolates, are actually the individual colonies. And what you see there are samples of water that were taken from the beach, the creek. This is in an old study that I did in San Diego. You see Enterococci from bird stools and kelp. And what we did was we were interested in looking at the potential sources of Enterococci to Moonlight Beach. So we did some speciation work to look at the actual distribution of the different Enterococci species in these different samples. So in the bar graph, what you see is the percent isolates, that's on the left. The bottom shows the different types of samples that were analyzed, and below that you can see the different species of Enterococci and the color that matches what you see in the individual bars. So basically what you're looking at is a pattern of the species distribution in each sample so that you can compare across sample types. You can see on the far right, this is the distribution of Enterococci species that was found in beach water, which looks very different from what we saw in the creek flows. So this is what I mean by doing further characterization and using speciation. Unfortunately, that's not something you can do using IDEXX. And I won't go into that into this talk, but it's very challenging. All right, so those are the two basic approaches for quantifying fecal indicator bacteria. Now I want to talk about the limitations. And that is that the counts can be under or estimated by both approaches. And the results can vary depending on the water type and sample location. For instance, in brackish water, there may be constituents that cause autofluorescence, for instance, that could lead to a false positive reading. The results can also vary by method and by volume of water tested, and I'll talk about that in a little bit. Interestingly, there was a presentation done by the city of Santa Cruz at our Northern California Beach Water Quality Group meeting. They reported that IDEXX CoAlert overestimated the total coliform counts for 6 beaches that were on the 303D list. And it turns out that the county of Santa Cruz was using the IDEXX CoAlert method, and the city was using membrane filtration. And the counts actually differed between methods by up to 2 log, which is significant. Other studies have also reported false positive results using CoAlert for marine water. Now I'm saying this is what they found at those particular beaches. That might not be the case for your beach. as I mentioned, the volume of water tested matters. In most cases, the greater the volume tested, the more representative the count. The counts are reported per 100 ml of water. So if you're testing low volume, say 1 or 10 mLs, this can lead to overestimating the counts due to extrapolation errors. Okay, another limitation is, and this is for membrane filtration, is that filtering high volumes of turbid water samples can lead to underestimating counts due to filter clogging or masking of the bacteria. Okay, some more limitations. Insufficient shaking of samples prior to testing may lead to inaccurate counts, and this is because the bacteria are not homogeneously distributed in water samples. They may be attached to other bacteria, cells, and particulates, so shaking very vigorously is important. Now, this is something interesting that many of you may not know. You get different, you can actually get the same count using different volumes of water. So for instance, if you take 100 mLs of a water sample, that may yield 15 total coliforms. But then if you take an aliquot of that, of 1 to 10 dilution of the same sample, that may also yield 15, oops, that was supposed to be total coliforms, sorry. So you can get the same count using different volumes of water. So what do you do with that? Well, I personally would go with the larger, I would trust the results from the 100 ml versus the 10 ml, but you can see that these discrepancies happen because the bacteria are not, homogeneously distributed in water. And then both methods, this is important to know, that both methods will detect bacteria that are not considered total coliforms, fecal coliforms, and Enterococci. So, in other words, you can get false positives using both methods. We know that using IDEXX CoAlert, you can detect Vibrio species, which are not total coliforms, and with Enterococci, we can detect many closely related bacteria that are not Enterococci. All right, so why do we get different FIB counts using IDEXX and membrane filtration? The main reason is because bacteria grow differently in broth or liquid versus agar. And those are two different types of culture media that we use to grow bacteria. So for membrane filtration, that's an agar-based method. IDEXX is a broth-based method, so that accounts for some differences. The other reason for differences is because the units of measurements differ. In membrane filtration, we're actually counting colony-forming units per ml of water, so we get an actual count. I mean, basically, what we see is the count. With IDEXX, you're looking at, the number of wells in a quantitray that are positive, and then using a statistical analysis to come up with the most probable number. So that's the other reason why the counts may differ between methods. Okay, and then something that many of you who are not microbiologists may not understand is that not all of the FIB methods measure the same population or genus and species of organisms, or the same proportion of genus and species. So this can account for differences. And then, of course, there's the interlab and intertechnician variability and biological variability. So by biological variability, I mean we talked about the fact that there's not homogeneous distribution, so that when you're doing, say, counts from duplicate samples, those numbers may vary. Or even taking samples 10 minutes apart at the same site will yield different counts. So why are we still using fecal indicator bacteria? Well, the reason is because the methods are cheap and easy to perform, and there's been some epidemiologic studies that have shown a correlation between FIB counts and illness rates. But caution, even epidemiological studies have shortcomings. Results of the early epidemiologic studies were based on FIB methods that were actually less specific than the currently used methods. So, for instance, for Enterococci, we know that the methods used 20 years ago were very different from the methods we use today. And another thing is that across these different studies that have been done worldwide, the methods differ. So they can't be, they're not really directly comparable. All right. There's also been inconsistencies between epi studies based on, geographic differences where those studies were conducted. A review of recent epi studies found that Enterococci had the best relationship to health risks among the different indicators principally used for marine water, but less than half of the studies found a significant health relationship. And the dose-response curves establishing the relationship between increased illness and indicator density were highly variable. Recently, the EPA conducted a near-epi study that showed good correlation between Enterococci and illness rates, but the sites that were studied were impacted by point sources like discharge from sewage treatment plants with known human fecal contamination and combined sewer overflows, which doesn't necessarily represent beaches elsewhere, like in California. Now, more recent epidemiological studies conducted at beaches in California showed that there was no correlation between FIB levels and illness rates, and that may be because, unlike beaches on the East Coast or at the Great Lakes, beaches in California are impacted primarily by urban runoff and or natural sources. Okay, some of you heard about or have been hearing about proposed ocean monitoring regulations, and these are, the proposal is to use Enterococci and E. coli alone rather than total and fecal coliforms. And they're proposing using Enterococci for marine waters and E. coli for fresh water. These regulations are for water quality assessment and TMDLs. They're not for AB 411 monitoring. But I did want to comment on the potential limitations to using a single indicator. First, starting with Enterococci, and that's that because you heard they're widespread in occurrence and they are able to persist in the environment, so this may actually confound assessments of fecal contamination and remediation efforts. That's a huge limitation. If you've got fecal coliform or E. coli results, you can get more information by comparing those results with the Enterococci counts. And that's because Elevated Enterococci levels can occur in the absence of fecal contamination due to natural sources such as plants and soil, etc. And let's see, where was I? Okay. So just because they're present in the environment doesn't necessarily indicate fecal contamination. So, but if the Enterococci counts are elevated and the E. coli or fecal coliform levels are non-detected or low, this suggests natural source inputs. But if the E. coli and fecal coliform counts are also elevated, in addition to Enterococci, this is actually more indicative of fecal contamination. So by using multiple indicators, we can gain more information. Now, so I just wanted to share a few observations based on years of monitoring FIB in Southern California beaches and stormwater. After sewage spills, the counts for all three indicators are high. At sites that are impacted by high bird densities, total coliforms and fecal coliform counts are generally higher than Enterococci. In stormwater or from water bodies near aquatic vegetation, it's not unusual to find Enterococci counts higher than fecal coliform and E. coli. Consistently high Enterococci counts suggest plants or soil may be primary contributors. However, fecal waste for animals that consume plants must also be considered. So the bottom line is the more indicators you use, the more information you can derive regarding sources. All right. Some considerations for assessing the swimmability at beaches given these limitations. First, know your beach. Ask these questions. What are the potential sources of fecal contamination? What are the typical background levels of fecal indicator bacteria? So, because most beaches in California exceed the Enterococci standard, you need to look at your background levels. Are they near or above the water quality limit? That could be suggestive of a constant source of Enterococci. And because they're near the limit, are the increases in counts due to random fluctuation or biological variation? Another consideration is, could the variability in the FIB counts reflect changes in test methods, testing lab, or testing personnel? And then think about IDEXX. Is that appropriate? Is that the appropriate method at your beach? You've got to know what organisms your test is detecting. What's your false positive rate? What's the proportion of fecal indicator bacteria counts that reflect non-FIB or environmental strains? Okay, so that is that's the end of what I'm going to be talking about. I want us to move forward and talk about Enterococcus qPCR. So for that, I'm going to turn it over to Joe Guzman.
Okay, so thank you, Donna, for asking me to help you co-present this talk. I know we're short on time, and we wanted to leave some time for questions, so I'll try to get through my slides quickly. This is the overview of what I wanted to cover as far as Enterococcus qPCR, and then just the rapid molecular methods in general. So obviously, any regulatory testing requires the use of an approved method, so I'll briefly talk about the Enterococcus PCR as an approved method. I'll do a brief overview of the qPCR. I'll talk about the advantages and disadvantages, and kind of in the framework of the title, this is where the good, the bad, and the ugly kind of come in. The advantages obviously are the good, the disadvantages are bad. And when I was trying to think about ugly, I don't know if there really is an ugly, but we'll kind of bring that up maybe at the end of the talk. qPCR has been approved for use since 2012, and so it's been 5 years. And has anyone implemented the use for routine monitoring? I'll give some examples. I'll put out there just how many labs are capable of doing qPCR in California, and if we ever decide to go down the path of implementation, what labs are capable. And then I'll discuss what I think are the practical uses for qPCR for our lab in Orange County. So Enterococcus, qPCR is an approved method. Obviously, EPA in 2012 put out their recreational water quality criteria. This basically, on the federal level, approved the use of the Enterococcus qPCR, and I've listed the standards that they put out. And then also EPA, there are two EPA methods for Enterococcus qPCR, EPA method 1611 and then EPA 1609. And what EPA recommends is EPA 1609 because it incorporates a internal amplification control. And obviously, of course, you can't just have the federal approval. California also has to give its blessing in order to run these methods. And so in September 2014, Senate Bill 1395 was signed. And so basically California approved the use of EPA 1609 or EPA 1611, but only after side-by-side testing over a beach season shows that the results are comparable. And actually, I recently heard that the state lab is planning to do a collaborative study with San Diego County, do a year-long study actually doing culture compared to the qPCR validation. Just a brief overview of qPCR. In this slide, I basically took from Squirp. This was a two-page fact sheet that Squirp has on their website. I slightly modified it to just focus on the part I wanted to discuss, which basically is how qPCR works. Obviously, we want to try to use these methods because you can get your result in 2 to 4 hours versus the 24-hour culture methods. And how does it work? Basically, DNA replicates itself as part of many normal biological processes, and the qPCR imitates this process but speeds it up in the lab environment. So step one, basically bacteria in the water sample are captured on a filter and mixed with reagents that simulate normal DNA replication. Step two, your fluorescent probes that adhere to the new DNA copies are added, and the sample is subjected to repeated heating and cooling cycles to double the amount of DNA every few minutes. And then the third step, computer records the amount of fluorescence in real time to estimate how much DNA and how many cells were present in the original sample. So Enterococcus qPCR, the good, it's our advantages, rapid, so there is a potential for same-day results, but trying to get results by noon, which is the target, could be very, it's doable, but you have to have the right conditions to be able to get that result out by noon. The assays are specific, so your assays can be designed to target very specific organisms with minimal to no cross-reactivity. And the nice thing about these assays is when the technology advances and even more specific methods come out, it's not that difficult to adapt to the new version. And then the correlation with culture methods, as we mentioned, in most of the epi studies, correlation has been fairly good with culture methods. The disadvantages, cost, that's a big disadvantage. Complexity of the test, standardization, the false positives due to DNA contamination, results may not be available until afternoon, precluding same-day warning to swimmers, and then the lower throughput of samples tested per day compared to IDEXX and membrane filtration. So I'll talk about the correlation with methods. Most of the comparison studies done in the last 10 years show good correlation between qPCR and culture methods when you have positive samples. When you have samples less than the cutoff or less than 100, then the correlation between culture and PCR is not so great. But when you have positive samples, samples above the cutoff, then you see better correlation. And some reasons for the discrepancies, and when you're looking at qPCR results, it tends to overestimate. And one of the reasons is you're measuring different endpoints. The culture methods, they detect viable organisms for growth, and your qPCR detects amplification of DNA from viable and non-viable organisms. Another reason for maybe overestimation by qPCR is the calculation method. The EPA method recommends a delta-delta CT calculation, and in other papers or studies you'll see that a delta CT calculation is more appropriate. And then one reason where your results might not agree, or an underestimation by qPCR, would be inhibition. So something in your sample that's inhibiting the PCR reaction, so you could get potentially a false negative result. So now I'll switch and talk about disadvantages. One is the cost. So I just want to mention a disclaimer. Our lab does not endorse any of these products. I just want to put that out there. These are just the ones we happen to be using. And then the costs are just our estimated costs based on what we had to pay for these items. And then when you look at the per sample cost, that only includes labor and consumable supplies. So, cost, a ballpark figure, in our lab, $65,000 for a Step 1 Plus instrument, and then $135,000 if you're doing a more sophisticated ABI 7500 instrument. And so a range, I feel comfortable putting out a range. If you go in with, there are some real-time instruments that you can get for maybe $25,000. So maybe your startup cost for equipment might be in the range of $40,000 to $50,000. And then on the high end, so for something like a droplet digital PCR, your cost could go to about $150,000. So I've listed the costs for some of the things, the centrifuges. You need a microcentrifuge and some mini centrifuges, bead beaters, vortex, pipettes. $6,000 to $7,000 for pipettes might seem a lot, but you have to have separate pipettes for each workstation. And if you can get an electronic pipette, that just helps with your accuracy, because in these molecular methods, pipetting is a big deal. You need to have accurate pipetting to make sure your results are good. And then some labs can invest in a PCR workstation, and the cost for that $2,500. And then when we did our fee study back in, this is 2010, costs, one target is about $85, and then for each additional target. So the nice thing about doing the molecular methods, once you extract your DNA, then you can test that extract with multiple targets. So if we have to add an additional target, then the cost is about $37 for that additional target. And then another disadvantage would be complexity. The recent Byte 13 report that just came out, technology transfer in the Byte 13 SQUIRT trained 14 water agencies to perform Enterococcus qPCR and HF183 qPCR. So in the short term, the labs maintain proficiency through the intercalibration study. So, you know, it took maybe a couple weeks to train the labs on the assay. They gave them a month or so to practice it, and then they SQUIRT challenged each lab with a validation panel, and most of the labs, I believe 8 of the 14 labs, were able to pass that validation panel. So, you know, if you're routinely running the assay, you can maintain your proficiency. But in this study because most of the sample collection occurred in 2013, 2014, labs weren't running the actual PCR assays until 2015, 2016. So, you know, they got the original training and were running it routinely in 2013, but when they went to then process the samples in 2015, 2016, the study showed that the labs didn't always maintain their proficiency. So if you're not routinely performing the molecular methods, then it's really difficult to maintain proficiency. And then what I think is the major disadvantage is the standardization. So the current EPA method relies on a standard curve to then derive your quantitation from your samples. EPA method provides procedures for labs to make their own DNA standards. So the DNA standard concentrations can vary from lab to lab, and if you have this variation, then it's hard to compare your lab results to another lab. And then even if you get one vendor to provide all the labs the same DNA standards, it's still difficult to replicate the standard curve from one lab to another. And the picture has I just pulled this off the internet of maybe a perfect, this is a perfect standard curve, but in real life, it's really hard to achieve this, and it takes a lot of practice to get a good standard curve. And basically, the standard curve is the tipping point for whether your quantitative results are going to be accurate. So if you can't replicate a good standard curve, then your results will be questionable. So now switching to qPCR implementation. Again, like I said, 2012 qPCR was approved by the EPA. In 2016, the EPA had the Recreational Water Conference in Louisiana, and these were some of the presentations given to what were these labs' experience in implementing qPCR. So Michigan, as far as I know, Michigan probably is the only state that implementing qPCR on a statewide basis, and they're using E. coli qPCR because for the Great Lakes they're dealing with freshwater versus marine water, so the standard for freshwater is E. coli. So they got a bunch of money. They invested in equipment to then place at 16 labs. They trained each of the labs to run this E. coli qPCR. From what I understand, that's where they're at. I'm not sure how they've progressed, but the thought was once the labs are up and ready, they would run their samples, they would send their data through a data portal, and then the results would get posted to the public, and hopefully to get same-day results. Another example in Chicago, where they compared Enterococcus qPCR to E. coli culture at 5 beaches. Basically, the outcome for this was the culture did not correlate to the qPCR, and in fact, they still would have more postings based on the culture method compared to the molecular methods. And then another example in Hawaii, when they tried to implement, they did Enterococcus qPCR at 12 beaches. The problem in Hawaii is they were processing maybe 130 samples, and of those, 69% of the samples were inhibited. And EPA does have a protocol for how to deal with inhibited samples, and so when they applied that protocol to see if that got rid of the inhibition, still 60% of those samples showed inhibition. So, this slide kind of shows that if you're going to try to implement it, you really need to understand your watershed, because the molecular methods are probably not going to work in all areas. And then just wanted to mention which labs in California have Enterococcus qPCR capability. So 8 labs in Southern California were trained to do qPCR, and I list the 8 labs. I'm sure there are more university labs that have capability, but I just listed the ones in Southern California that would potentially do routine beach monitoring. And then I'm not so familiar with the Northern California, but I know the California Department of Health Drinking Water and Radiation Lab in Richmond has capability to do the rapid methods. I think they're the ones tasked with trying to validate the EPA Enterococcus methods for routine use in California. And then Humboldt County in Public Health Lab, I knew they do a lot of shellfish testing, qPCR for Vibrio, and I just saw an email where they're trying to implement the qPCR for the gull marker. And so I'm sure if you can do the gull marker qPCRs, you definitely can do the Enterococcus qPCRs. And then, so practical uses. So like I said, it's been 5 years since EPA has approved the qPCR for routine monitoring. For us, I just don't think it's practical to test routine, to do qPCR for routine beach monitoring. We have, a total of 150 sites in Orange County that get tested once a week, and to try to run those routinely, I don't think it's going to happen. So maybe a practical way would be to select sites with higher rates of exceedance, and this maybe use qPCR to get a quicker result. And then also the other thing is, when you're using the rapid methods, it's recommended to test the sites more than once a week. And so, without, getting rid of some of the other indicators that we have to test for, I don't know if it would be cost effective to try to then do the rapid methods and not only do it once a week, try to do it more than once a week. So where we see the use more is in for post-exceedance recheck samples to reduce turnaround time to unpost the beach. So we have a culture result that is exceeding the standard. Our samplers will go out and collect a recheck, and maybe if we run the rapid method on that recheck, we can unpost the beach quicker. And then the other application would be for sewage spills. At this point, if a sewage spill makes it to the beach is closed. You need 2 negative samples to reopen the beach. And so if we could do the rapid methods on the sewage spill samples to get a quicker result, then you could potentially reduce your beach closure by probably a day. So, and then the other application for us, which is more reasonable than to do it for routine beach monitoring, would be for source tracking. So with that, I'm going to turn it back over to Donna for the future considerations and summary.
Okay, thank you very much, Joe. So we just wanted to leave you with some thoughts. Advances in science and technology will continue to result in the introduction of more complex, expensive, and difficult to interpret tests. New methods advance quickly, so we need time for rigorous review and improvements before implementing for regulatory use. New methods should be used as RUO, research use only, until proven and widely used. Widespread use of new methods often reveal limitations. A paper just came out this week by Alm et al. That showed that the HF183 human marker is present in bird stools. This is something we knew about in Orange County based on some work done by Dr. Ted Von Bittner and Joe Guzman at Doheny Beach. Be patient but vigilant. There's been pressure on regulatory agencies to approve advanced methods quickly, but the downside is that hinders or can hinder rigorous validation. So in summary, understanding the limitations of FIB and test methods is critical to making more accurate assessments regarding fecal contamination. Avoid FIB testing abuse. I love that. Test abuse. Focus on FIB counts or results based on a single indicator. Focusing on that will lead to further problems. It can be misleading. Verify questionable results. Identify the organisms being measured. Always correlate FIB data with source inputs in environmental conditions and also consider biological variation. Enterococcus qPCR can yield results in a few hours. It's useful for reopening posted beaches quickly, but then the results in data interpretation may vary between labs and beach sites.
Yeah. So if I had to add a ugly part to the molecular methods, that probably would be misinterpreting the data or maybe using a rapid method before it's been fully validated.
Okay, so that brings our presentation to a close, and I don't know if we have time for questions, Eric?
We do have time for questions. In fact, we've been receiving some by chat. Let me address those first, and then we can take questions from the phone. Donna, the first question to you, can Entero hibernate in the surface zone only to be activated when stirred up by dredge discharge or exposed to good, what media in the lab, I guess?
Okay, so I don't know of any studies looking specifically at that, but I can tell you that Enterococci are biofilm producers, so they can, in a sense, survive, or if you want to call it hibernate, for very long periods of time when they're in biofilms. And when those biofilms age and disrupt, that releases the Enterococci. We know there's biofilm on particulates like sand. So yeah, that's very possible. I just don't know if hibernate is the right term.
And we had a couple questions for Joe. The first was coming from San Diego, where they have a hepatitis A outbreak. Should they be testing creeks, confluents, and ocean waters by any one of these methods? Joe's response via chat was, it's difficult to test for the actual viruses in water because you need to test a large volume of water.
I added another response that maybe consider using human-specific coliphage, because the coliphages have been considered as a potential surrogate for viruses. So that might give you a clue that there could be those viruses in the water.
Excellent. Thank you, Joe. You had one more question coming through. You want to just take that one, Joe?
How has the EPA
program?
Yeah. Been using yeah, see, as far as I know, there really, I don't think implementation, qPCR hasn't been implemented for routine monitoring. It's just, anytime we've done it, I think there was only maybe 2 months in 2010 where we actually used qPCR results to do postings at the beach. But other than that, I don't know of anyone that really is using the rapid methods for routine beach monitoring. I think a better, maybe a more applicable, which has been used more widely, is probably the predictive modeling. So if you want to get a daily, FIB result or what's the water quality, I think predictive modeling might be a better way to go than the rapid methods.
Thank you, Joe. And for those of you that are still on the line, the webinar that will be presented in April is on modeling and nowcasting.
I see there's a question about using qPCR rapid method data for detecting elevated bacterial levels in the drinking water system. So that's a good question. There's a reason why drinking water systems don't use qPCR, and one of the main reasons is because it detects non-viable organisms. I mean, basically you're looking at DNA. So the fact that you find DNA doesn't necessarily mean that the organism is viable, and I think the concern is for false positives, and that's probably the main reason why it hasn't been implemented in drinking water labs.
The question is how we can all protect the public from any possible extreme bacteria or viruses.
That's a good question. And that's ultimately what we want to do, is to protect the public. And I think it really takes education, like this webinar, because people do panic when they hear that there's fecal indicator bacteria in the water. And we are doing it the cheapest and simplest way possible. In California, at our heavily used beaches, we use 3 indicators, which is a good thing. Actually, that is more protective than using a single indicator as being recommended by EPA.
Where to submit a recommendation for the future draft? I came to my own analytics research study how everyone can create greater resilience in their own body for any viruses or bacteria.
Gosh, I'm having a really hard time hearing you. Can you repeat that? Are you asking where can you present your study findings?
Yes, where we can present or submit recommendations to...
I recommend perhaps going to one of the beach water quality working group meetings. There's one in Southern California and in Northern California. If you look on the California State website under water quality, you can get more information on the meetings there. It's the Beach Water Quality Work Group meetings.
Thank you.
Donna, several times you've recommended using multiple indicators to identify contamination. Do you recommend using the same method, or do you recommend combining methods? Or, and if you do recommend using the same method, do you have a preference for one?
Yeah, so I'm biased. I like using membrane filtration, and it's just because we can, like I mentioned, we can do speciation, we can do DNA typing. But on the other hand, for high volume, we in our laboratory we do use the IDEXX methods, but we will question, the results if they don't make sense for the beach that we're monitoring, or if we continue to see exceedances, then we'll question those results, and we'll want to verify using membrane filtration. In the public health lab, we use PCR for identifying, pathogens that can cause disease. And if we get a questionable result using PCR, say a CT value that's very high or that doesn't match up with the clinical presentation of the patient, we automatically repeat the test and we test using an alternate method, which could be culture or non-culture. And this is something that's not typically done in environmental laboratories.
Great, thank you.